many papers, about more than 200, with 11 patents, and he's won many awards, including first prize from the Prime Minister of Poland, prizes from the Rector of Warsaw University of Technology for scientific achievement, and the Mieczysław Wolfif Prize. The floor is yours, Professor.
Thank you for your kind introduction, and thank you for invitation and for giving this talk. It's my great pleasure to be back in LA in Ali Khademissani Group, and I'll be happy to share some information about our results related to biofabrication, although I will focus a bit on microfluidic-assisted biofabrication. As was mentioned, I'm coming from Poland, far away, more than 14 hours fly, and I'm coming from Faculty of Material Science. So my background is material science and engineering. I did my PhD in biomechanics, so I have also some background in biomechanics and biomodeling. So I'm coming from Warsaw University of Technology. It is the biggest technical university in Poland and ranked #1 technical university in Poland. It is established 1826, so it's consisting of 19 faculties. Material science and engineering is one of the smallest faculty. We have 3 levels of education: bachelor, master, and doctoral study. Regarding faculty, it's starting 1929. Maybe you know Professor Czochralski. He was founder of our material science division, and he's the founder also of the method to single crystal fabrication. So currently we have about 400 undergraduates, 90 students, and about 40 academic staff. So as I mentioned, we are small faculty. Regarding to my group, it's consisting about 20 people plus master and bachelor students. So mainly interest, since we are biomaterials group, we are working on developing new materials for medicine generally. We're working on the 3D printing of different types of materials, composites, polymers, metals. We are also working, as I will be talking today, about bioprinting, biofabrication, and microfluidic systems. We have some work done on electrospinning, and of course, with advantage of 3D imaging techniques, we can prepare 3-dimensional geometrical models for rebuilding or mimicking natural structures. Some parts of my group are also focused on different drug delivery systems, especially we are developing 3D printing drugs or 3D printing coating consisting of the drugs which we apply to medical implants. So, sorry, something was wrong. I don't know what's happened. So yes, so we are working, for instance, on the fabrication of surgical models and templates for repairing mandibular after tumor removal. So as you see, there is a mandibular reconstructed with the bone, which was fabricated using our template. So we are working in close collaboration with surgeons. Regarding other application, we also have strong collaboration with Veterinaries surgeons. We are printing bone replacing materials like made of polyheteroketones. You can see dog working with these implants after again a removal of bone cancer. We also working with 3D printing technology, which we print 3-dimensional implants made of titanium, for instance. This is already applied also to the dog when we remove the bone affected by cancer. and then we apply this porous structure for bone ingrow implants, three-dimensional titanium implants. But today, we also talk about biofabrication, but before that, I would like to show some current application of 3D printing technologies. Again, this is metallic implants, titanium alloy. We develop new type of dental implants where you can delivery also drug to the cavity for better bone ingrow or for treatment on infection. So we have already a few clinical patients for preclinical study. So that's generally what we do with different materials. But again, today I would like to focus on tissue engineering applications, and especially when we apply different cells, growth factors, and scaffolds. That's different porous materials which can be not biodegradable, biodegradable, and which can be used for us. So I mentioned before for regeneration of different tissue or also as tissue models. So when we look for time scale of development of tissue engineering field, we can say that in the beginning, most work was done in 2D culture when we use only plates for making tissue-like structure, but was really simplified tissue. And since the 1988, we have already started looking for different three-dimensional scaffolds with different architecture to increase new tissue formation and to replace natural extracellular matrix for new tissue developments. And nowadays we are focusing on more advanced technologies with using 3D printing and additive manufacturing, where we can add also tissue functionalities in our three-dimensional constructs. So there are different technologies which are already present on the market where you can 3D print tissue-like structures for different application, brain, leg, vasculature, kidney, and so on and so on. So biofabrication, as you know, it's a process where we automatically generate biological functional products, which with structural organization from cells, bioactive molecules, biomaterials, and then we apply this to really mimic natural tissues, and for that we have different technologies like extrusion-based bioprinting. We have also a voxel-based bioprinting where we can apply selective stereolithography, volumetric 3D printing, or DLP for 3D printing of complex structures. But still, there are some people working on droplet kind of bioprinting, which can use inkjet, acoustic, or laser-assisted bioprinting. I will not talk today about the bioassembly. This is another field of biofabrication where you can assembly of organoids or some other type of micro tissues to form three-dimensional tissue-like structures. So since my work is focused on microfluidics, few words about this. You know that microfluidics is really growing field of science where we manipulate and control with high precision fluid in the range of microliters or even picoliters. And for that we use networks of micro channels arranged in different arrangements. So where we can use this kind of really simple methods, we can use this for organ on the chip, for instance, development, or lab on the chip generally. We can use in chemistry, cosmetics, pharmaceutics, and also in biofabrication. There was already work in 2016 published where people try to use microfluidics for cell encapsulation. So they use a cell and gelma, gelatin methacryl oil, and oil as some of the fluid use this microfluidic system. And by that, you can encapsulate in the drop cells. And they proved that when they encapsulate the cells in these drops, in capsules, that when they deliver it to the bone part, they have increased bone growth. So there are ways that you can use biofabrication using this microfluidics, where you encapsulate the cells and have microspheres which you can deliver to the patients. However, there are a lot of applications where you can use microfluidics for fiber fabrication. So you can use glass capillaries, simple glass capillaries, or made by PDMS chips, where you can fabricate fibers, continuous fibers with different architecture. It could be solid, tubular, porous, flat, hybrid, and so on and so on. You can even go to the approach where in one continuous fibers you can apply different types of cells and different types of bioinks, which are also important for biofabrications. So how we started with this microfluidics extrusion-based bioprinting? We have started with simple chip, which is allow you for quick coaxial extrusion bioprinting. So you have your hydrogel-based ink, and you have cross-linking in the shell. And by using calcium ions for cross-linking, you can quickly change from solid to gel situation. And then you can quantitatively assess the fidelity of your printing fibers, because you want to have good quality of the fibers, sometimes with diameters lower than even 100 micrometers. Why we want to high precision? Of course, it's nice to represent a high complex structure of natural tissue, but you can also be aware about oxygen delivery to the cells, which will be encapsulated in these fibers. Of course, there you can develop different strategies how to cross-link this pre-hydrogel ink. You can use, of course, dual cross-linking strategy, like many people are doing. So first, ionic cross-linking, then you use photo cross-linking, applying different photocurable polymers, even natural-based one. So one of the applications we started with was cartridge. So we have tried different hydrogel as bioinks. We use alginate gelatin-based hydrogel, alginate gelatin, hydronic sulfate, and alginate gelatin hydronic sulfate, and also hyaluronic acid. And by changing composition, as you see, you can also change compression modules, so stiffness of your ink, and which you encapsulate the cells. And also rheology of the ink is changing by changing the composition of your bioink. So it was interesting how this different compression stiffness influences cells encapsulated in these bioinks. So first of all, we was of course looked at cell survival, and you see all the hydrogels based inks we provide us with environment for the cells which allow them to be alive, and this viability of the cells was higher than 90% after even 3 hours after printing, but also later on during 3D culture. And when you culture, it is interesting that you can find different relation between the stiffness of the hydrogel and composition of the hydrogel and different markers like aggregants, collagen 2, collagen 1, and collagen 10. Of course, that was our preliminary study, and we show, of course, for cartridge, very good production of algae aggregants and collagen 2. However, we have seen also, especially for this hyaluronic acid hydrogel, which had higher stiffness comparison to other hydrogel, we had higher production of also collagen type 10, which is characteristic for this hypertrophic condition for the cells culture. So when we look for mRNA expressions, we've seen that the higher collagen 2, collagen 1 gene expression was presented for hydrogel with gelatin and chondroitin sulfate. So that was the best one for our application. But when we look for this collagen 2, collagen 10 gene expression, we've seen again what I presented before, that with hyaluronic acid, where you have higher compression stiffness, you have higher also collagen 10 expression. So that's quite interesting for us. So microfluidic extrusion-based strategy allow you to fabricate different structure using quite simple microfluidic chips. So you can imagine that you can add 1, 2, or even more bioinks to one chip, and then using some controlling of the pumps, you can deliver 1-to-1 or one up in one single fiber. You can deliver 2 different inks, or you can generate stacks of different inks and different even cells and different cells tissue-like structure. With adding some microfluidic plastic mixers, we proved that we can, using some also computer modeling, we can do gradient structures by controlling flow of different bioinks. So this is example where you can have in one fibers 2 alive type of cells, fibroblast and myoblast C2, C12. So this was some preliminary study for project related to muscle regeneration. And coming back a bit to the cartridge, since we was surprised that we can somehow control hypertrophic zones formation in the cartridge-like structure. So we decided to make a biphasic structure consisting of allular structure, articular surface, and a calcified zone of our cartridge. And for that, we developed 2 different bioinks. For this upper zone, we use again GELMA, chondroitic sulfate, as I mentioned before. But we also mix 2 types of cell, articular chondrocytes and also MSC, which was proven that this enhances cartridge formation. And for calcified zone, we was using additional hyaluronic acid to increase the stiffness of the bioink. We use 3 calcium phosphate nanoparticles as to deliver to the calcium zone some calcium ions already, which are also present in natural calcified zones. And we also apply this time only MSC cells. By this microfluidic modified chip with 2 layers, we was able to fabricate this hybrid structures. So this is confirmation that the chondrocyte stained in blue are only in the upper zone of our structure, and in the calcified zone we have a nice distribution of free calcium phosphate particles for better also bone with better integration with subchondral bone. All the cells was alive, of course, after printing process, what is the first evaluation tests. Of course, we look for expression of different markers to make this story short. We can confirm that addition of chondrocyte had a positive effects on expression on chondrogenic markers when compared to only cultured with MSC. And when we introduced free calcium phosphate and hyaluronic acid, we have caused overexpression over all proteins, especially collagen 10, what we expected to have. However, other expression was not so pronounced like in the case for this chondrogenic expressions. So what was our next step? We was thinking, okay, why we cannot, however, how somehow improve the speed of and alignment of the fabrication of tissue-like structure, where you have aligned fibers, especially for tendon and for the muscle, because then you have really naturally aligned fibers like structures. So for that, we develop bio-wet spinning, where we again use microfluidic technology to fabricate fibers, but the fibers was collected on the rotating drum. And by collecting them on the rotating drum, we could also, by changing speed of rotation of the drum, you can also change the fiber diameters, but also what is possible, you can also arrange them together, the fibers, making some kind of rings with aligned fibers, like you see here, with the cells in each of the fibers. So having such rings, which can be fabricated again by using GELMA and alginate for fast cross-linking, you can have fibers with diameter down to 100 micrometers. Why these rings are so interesting? Because then you can apply these rings in a mechanical static, for instance, stretching device or dynamic device. And we applied also dynamic static mechanical stretching together with biochemical stimulation, growth factors, BMP12 for tendon regeneration. Again, although we apply this drum rotating system, we have all this 94% of cells alive, so viability was very high, and the growth factor, growth of the cells were also high. Of course, when you add BMP12, the proliferation is a bit slow down. However, mechanical transduction promotes cell growth. And we look for expression, protein expression, especially for collagen type 1 and collagen type 2, characteristic for the tendon. And what was interesting, of course, they was secreted very well in whole fibers, and they follow the fiber orientation, so they was aligned along the fibers. And this also allow us to look for tenomodulin expression in such kind of structures, and tenomodulin expression was the highest for the ring, which are where we apply 15% strain and BMP12 addition. So this biochemical stimulation together with mechanical stimulation induce some synergistic effects for tenogen gene expression. So that's again some application. So could we use also for the muscle this technology? Yes, we can. Together with my colleague Marco Costantini, we have done a nice study where we use this time also the drum for collecting fibers, which was made of alginate and also PEG fibrinogen. And we use this for muscle regeneration. So when we apply skeletal muscle progenitor cells, about 2 × 10⁻⁷ cell per ml, we was able to see that after already 3 days, but really after 15 days, we have already cell alignment along the fibers, and we've seen a very nice formation of the myosin in the fibers, which also show that cells, multinuclear cells, are aligned along the muscle. So it's like muscle fibers, which was in our case also fibers of hydrogels. So we have also obtained some spontaneous contraction. I don't know why this movie is not. Oh yes, it's working. So already after 15 days of culture. So that's proof in the vivo, but we also vitro, but we also wanted to see how this works in vivo. And this is a model with reconstruction of the muscle in the tibial arterial muscle. And when we compare a control without our construct and the grafted construct with the cells aligned in our fibers, we've seen already after 20 days that our graft regenerated muscle part was significantly higher, bigger than control one. So that was proven that biofabrication assisted with microfluidic system, where you have aligned fibers, simulating fibers of muscles, allow better regeneration process. So how we can proceed next step? So what we wanted to do, we wanted to fabricate core shell fibers. Before I presented you bulk fibers, but we was interested in to use also core shell fibers for skeletal muscle vessels or muscle tendon junction regeneration. And for that, we developed another system, which, as you see, it's differs than before one. We have still drum, which is rotating, and we still collect the fibers on the rotating drum, as you can see. So you can also make kind of rings or continuous layer of the fibers. However, in this application, we can use core shell structures of our custom-made microfluidic chip. So you can have shell hydrogel and also a different hydrogel for the core. This allows us, especially for the muscle and vessel fabrication, that we can use in the core, not restricted to short-thinning behavior bioinks, because mostly in extrusion-based biofabrication, you have to have inks with short-thinning properties. However, in this case, we was able to apply even Newtonian-like fluids in the core, since we have a shell which keeps them in the place. So how we modify the fibers, kosher fibers, of course, by changing technological parameters, process parameters. So you can develop fibers with different diameters, but you can also play with shell thickness and the same, of course, core thickness, you have shell higher thickness or shell smaller thickness. Why it is important to control the thickness? As what I mentioned before, and when you have encapsulated cells, it's nice to control a diffusion of the oxygen to the cells through the shell. So you have to play with the thickness of the shell. If you have too small thickness of the shell, then you have too fragile fibers. So the mimicking of natural structure could be problem. So, but changing, as I mentioned, and process parameters like rotation of the drum and also fluid flow, like for the shell, hydrogen flow for the shell, for the core. In your microfluidic system, by this you can control, again, as I mentioned, shell and core thickness. Of course, with the higher thickness of the shell, you can apply different cell types. If you have, for instance, muscle-like cells and vascular shell-like cells, you can have mimicking natural vessels structures. So where we apply this, we try, of course, for the application we have already have some experience with muscle. So we was trying to see if we were using this core shell fiber for muscle reconstruction have some influence. So for this, we use again this system where we apply Myoblast C2C12, and also for the core we could use simply fibrinogen ink, which is low viscous materials where the cells can easily survive. And for core we use again alginate to have for the shell. Sorry, we use alginate to have better stability of the fiber. So again, you can see that we can develop core shell structures with aligned fibers. And so how cells behaved in the core shell fibers? We look for, especially for the core, because cells was encapsulated in the core. You can see some shell thickness. And after 14 days, you have already alignment of the cells, and the cells even better aligned since the fibers have smaller diameters than in previous study we presented. So again, after already 14 days, you can see some spontaneous contractions of the cells. How looked when we look for the cells about the differentiation? You can see a really nice actin, which also follow the fiber which was fabricated using this system. Myosin heavy chains also somehow was very good expressed, and when we look for cell orientation, very good cell orientation due to encapsulation of them inside the core of the system. And we apply to this, like previously from the tendon, here we apply also to our fabricated rings. We applied static stretching, dynamic stretching, 5%, 10, 20%, and we also apply electric stimulation in our custom-made system, and also combine this static stretching and electrical stimulations. So for all this application, we have high cell viability. And we was interesting how this different stimulation modulate myosin heavy chain isoforms. And what was very exciting that different stimulation differently stimulate this myosin formation. And for instance, when we look for myosin heavy chain 1, we have major overexpression in the case of electrical stimulations, how you see here. And when you have when you have dynamic stretching, you have again major overestimation of myosin heavy chain second. Sorry, I have to take some water. And when we have simply static stretching, we have combination of this overexpression for all isoforms of the heavy chain. So that's quite interesting study showing that applying different stimulus, mechanical, chemical, or as I previously also mentioned, biochemical stimulus, you can control regeneration process of in vitro of the tissue using biofabrication process. So finally, as I mentioned, we are processing with this information we have obtained for the muscle and for the tendon. Now we would like to use this for muscle tendon unit, so muscle tendon junctions, somebody told us. Yes, so then we are trying to apply this technology of core shell, since we can easily control composition of the bioink, which are important for, as I mentioned, for cells differentiations, of course, for proliferation as well. So again, using this system like I presented before, you can apply different bioinks and different cell types for tendon and for muscle part of this muscle tendon junctions. And for fibroblasts, you can use, for instance, gelatin-based, and myoblasts, you can use fibrinogen-based bioinks. And for our approach, we use fibroblast for the tendon and C2C12 myoblast for the muscle part regeneration. So using this controllable system, as you see on this slide, you can control alignment of the different inks, also different cells. You can have different arrangement of these fibers, and you can also have, for instance, have fibers we made of one type of ink and other fibers of different type of ink. And of course, we are also interested in what happened on the border between one and another bioink, because this is our muscle tendon junction, junction part. So of course, again, we have to do a lot, and Marina Popi, my PhD student, was working with Alessia for looking for good spinability window. So I mean, what should be drum rotation speed and ink flow rate to have good fiber shape, good fiber diameter for this application, and also ratio between shell and core diameter. And for this, as you see, there are different approaches that you can control. For instance, speed of the drum, that you have higher this transition, smaller transition zones. When you have smaller speed, you have higher transition zones for this muscle tendon junction. So you can see that by even controlling some speed of the drum, you can also make longer or shorter transition zones for these two tissues. And of course, again, applying this technology allowed us to fabricate, align, and alive cells in the fibers, as you can see here, by nuclei orientations, and also for actin orientation. You have the cells align according to fiber long axis. And of course, this also depends a bit on the speed of the drum, but not significantly. And also the same was done for Myoblast, and we again achieved this Myoblast line and alive and secreting proper ECM markers proteins. So that was our proof of concept, where you can see that in one ring you have part where you have only muscle-like structure. You have again mixture of these two type of cells, and then you have tendon-like structure with also aligned fibers. And to speed up even process of this muscle tendon junction, we have developed another microfluidic chips with multi-channels. As you see here, with these multi-channels, you can simply fabricate faster and also more precisely alignment of different fibers consisting of different bearings and cells. And with that, definitely we are expecting to speed up process of biofabrication of these junctions. So let me go to conclusions. So after what I said, somebody can conclude that there are two things which we should look during fabrication. Of course, proper bearings. I didn't say too much about this today, but please look on the proper bearings. There are different approaches, mostly are natural-based bearings. There are some extracellular matrix components which are applied in bearings nowadays, but the most important that you can control the properties and that you know what kind of bearings you are using, what kind of interaction they present with the cells, especially in the time and especially in different conditions of the culture. And another point which is important, which was mostly my presentation about, is this biofabrication technologies. As I mentioned in the beginning, there are different ones. Some people are focused nowadays on this volumetric 3D printing to speed up process of fabrications. However, still, I think this microfluidic-assisted bioprinting allow us to really reproduce and mimic some of natural structure with very high precision, which is not possible using other technologies. And what is also important, we can also use this technology to do this very fast way. What is also important for the cells, not to print them in the environment of the 3D printing for long times. Of course, future studies will have to focus on, of course, on developing different things, which like a natural ECM change in the time. So this kind of 4D ECM mimicking constructs instead of using this passive scaffolds, passive ink, which degrade because of hydrolysis or enzymatically. We have to add some components to allow cells to control environment during tissue growth. So with that, I would like to acknowledge my collaborators, of course, Ali Khadamesseni. He's not anymore on UCL, sorry. Ali, that was, yeah, my mistake, but also my collaborators from Poland and around the world. And I would like to thank my group, which have been all this work, and especially the people working in the muscle, like Marina, and also with the vessel Alessia, but also there are people working in the three-dimensional cancer model, like Eva, drug delivery system, David, three-dimensional micro CT, Carol, and other people which have been to develop this 3D printing technologies. Thank you very much for your time, and I'm open for some questions.
Thank you for the great talk, Professor Wojciech. Appreciate it. A couple questions are, what is the next step for your research? Are you going to explore making these cartilage or other tissue constructs for organs on a chip, disease models, healthy models, etc.?
Thank you very much for the good question. Of course, as I presented in the beginning of my presentation, we would like to go as possible to the clinic with our application, but you know, probably better than me, that is very long way. Of course, tissue engineering could be really still has some big potential for regeneration of the missing tissues in the body, like bone, cartilage, especially muscle part, as well as we presented for the animal model. However, going to the clinic, to the patients is especially when you apply the cells in Europe is very difficult. So definitely looking for tissue like structure for in vitro testing of different drug and diseases are our also aim. As I mentioned, we're developing vascularized three-dimensional bone cancer on the chip to study extravasation of the cells. So that's definitely of course the future application for our work. Although, as I mentioned, our dream is, of course, to help directly patient in for regeneration of the missing part of mostly musculoskeletal tissue.
Thank you. Another question is, you introduced core shell fibers. Are you going to explore like making vasculature or vascularized tissue type of things? A lot of people are pursuing those.
Yes. As I mentioned already, there was not time to present. We are almost ready with a submission paper with Alessia for using this core shell, of course, for vasculature. So that's our aim, of course, because vasculature is core shell structure. So definitely this really represent, and you can really control the microvasculature and macrovasculature depend on the parameter of the process of fabrication core shell structure. Yes, thank you for this good question.
Thank you. You mentioned in the beginning about microfluidics extrusion-based printing, and then you moved into wet spinning. I was a little bit confused about, are you using microfluidics and wet spinning? Is this a method you invented? Can you a little bit elaborate? Because I heard about electrospinning, but this wet spinning and microfluidics, can you elaborate a little bit more?
Okay, of course. Maybe I'll use a slide to maybe it could be this as well. So maybe, because when you say about wet spinning, generally, so you extrude some fluid to coagulation bath, which allow you for cross-linking. So this is not different that we are doing here. We're extruding a precise coarse fiber to a coagulation bath, which consists of calcium ions, and by that you have this kind of wet spinning system, because you collect and drum your fibers after this after reaching this coagulation bath. Is understandable now?
Mm-hmm. Yes.
Okay.
Thank you. One other question is about slide. I don't know, you had a slide about spinnability window. What do you mean by good spinnability? Also, aren't you concerned about the bioink parameters or whatever the solution for the spinnability?
This one, maybe?
You had a heat map about spinnability. Yeah, this one. What is good spinnability, bad spinnability? You talk about drum rotation, ink flow, but how about the bioink parameters?
Okay, so good spinnability. So again, you can have fibers of different diameters of shell and core, and we are looking for good kind of ratio between core and shell thickness for our application, depend on applications. And for that, when you have bad spinnability, you can have even not form fibers. Our fibers are not reaching the right diameters. So therefore, you have to control drum rotation and also ink flow rate.
And these fibers have cells inside?
This not. This for spinnability window, we are using without cells. And the cells not change too much. We are lucky. Of course, you can say, okay, it should change because the rheology is changing. But for our inks, we are using especially this low viscous ink for the core. This doesn't change too much when we apply the cells.
Thank you. Is the machine that you use for wet spinning, is it available commercially or did you make it homemade?
We made homemade, although we are thinking to sell this, especially to research labs. If you are interested, Mehmet, we can provide one for you.
Well, email came from the group, so I'm going to let them contact you later. What salt of calcium ions did you try? Have you explored the impact of different calcium salts?
Yeah, good question. Good question. Of course, calcium ions is especially when it's too much calcium ions. We know that especially for the muscle application, is also not good. So we definitely optimize as low as possible calcium concentration in the calcium bath. 2%, even lower, to have, of course, still good feasibility of your construct. However, we don't want to have too much calcium because there are some, of course, calcium left in the fiber, but low amount. Very good point, Marat.
Thank you so much. That's all the questions I have. Thank you for the great talk, Professor Wojciech.
Thank you very much again for your kind invitation, and I hope to see you soon. Bye-bye.
Thank you. Bye-bye.